Hello, everyone. Welcome to our lecture number 4 on single-cell high-throughput data science lecture series. Today, we are going to talk about dimension reduction and data visualization. I will introduce several dimension reduction algorithms like PCA, tSNE, and PHATE, and see how they can apply to single-cell RNA-seq data visualization. So from the last couple of lecture series, we know from a single-cell RNA-seq experiment, we can get the expression matrix, also called the UMI count matrix, for genes and their expression values in multiple cells. So here we have a genes in the rows, and then you have cells in the column. Okay, so that's our starting point of our analysis. Remember last time we talked about the normalization and normalized by the library size. So there are procedures you can make this library size to the constant values for each individual cells. And after that, we are going to start from this normalized expression matrix and go to the next step. Will be visualize the cells in a 2 or 3 dimensional space. Okay, here is a standard workflow or pipeline you are going to use to do this single-cell RNA-seq data analysis. So we have the expression matrix, and then through the dimension reduction, you will project each individual cells in this 2-dimensional space. If those cells are similar to each other in their gene expression profile, they will be projected to a space close to each other. So this gives you a foundation to do the clustering analysis. In this example, you cluster cells into different groups, and then from there you can identify gene expressed specifically among particular clusters, but not others. And then you can use those information to annotate the cells belongs to the same clusters. So that's the basic workflow of the single-cell RNA-seq data analysis. So the key steps of this will be that's step 1. Start from the gene expression matrix to the embedding the algorithm to reduce the dimension and project the cells into this 2-dimensional space. So this lecture will focus on how are you going to do this dimension reduction, how are you going to project them onto this 3 or 2-dimensional space. So that's a very basic step. That's basically that's step 1 of the analysis. Let's start from an example. We have a UMI matrix. And if you know this, each row is a gene, and then each column is a cell. So that's what we know from the previous lectures. So now I'm going to do a transpose of this matrix. Essentially, I will move the rows into the column. So just rotate this matrix, transpose it. And after that, the columns will become genes, gene 1 to n, and the cells will be in the row cells. So you don't have to do this, but in data science, people normally put your samples in the row and your features, which describes your samples, as a variable in the column. So I make this transpose in order to use convention in the this convention in the data science. So people know we are going to classify to project cells, which rows instead of the genes, onto the low dimensional space. So that's the result of my transposition. After my transpose the original UMI matrix, now I have cells in my rows and I have genes in my columns. So now we have to have an understanding of the dimensionality of your data. So you can define the samples, in this case as cells, using either 1 gene or 2 gene or 3 gene or more. When you use 1 gene, you separate or describe your cells using one-dimensional information. That's indicated here. So you can have a distance between cells. Once you have 2 genes to be taken into account, then you are talking about use 2-dimensional data to describe each individual cells. If you use 3-dimensional 3 genes to describe these cells, then you are talking about a 3-dimensional data space. In the teaching of the, I realize some of the students have a difficulty to understand this dimensionality. So I'm going to give you examples, the real examples, to illustrate my point. So for example, you have this huge matrix, but now you only consider 2 genes. Okay, although you have maybe thousands of genes can be used to describe each cells, but here we only consider two genes. That give you a 2-dimensional description of each individual cells. So if you plot those cells position in 2-dimensional space, see here, the x-axis is the level of gene expression level of gene 1, and then the y-axis you have a gene expression level of gene 2. Now I'm going to put each individual cells on this 2-dimensional space. The first cell I have a gene expression 1 as 8. So I'm going to put number 8 here, and this have a gene expression level for gene 2 at 15. So we can find the coordinates for the cell number 1, which is located here. And then similarly, we put the position of cell number 2 on the space, which is 2, 1. So we have 2 here and 1 here. So we have cell 2 is located here. And then for cell 3, similarly, you have a 1, 6. So the position is 1 for first gene, 6 here. So that's your cell number 3. Okay, so for this example, you don't have to reduce any dimension because we only use 2-dimensional information to describe each individual cell. So you can do directly on this 2-dimensional space to find the position of each individual cells. Say 5, 0, 5. this will be C4. 1, 6 will be the same positions. C5, 2, 6, cell 6, 4, 9, cell 7, 2, 5, cell 8. Okay, so I'm done and plotting the relative position of the cells. So you can see cell 3, 5, 6, 8 have a similar profiles, and they are close to each other in this two-dimensional space because they have a similar profile in terms of the gene expression of these two genes, and they can be clustered together. Okay, so that's two-dimensional. So now if we decide to consider 3 genes expression instead of 2, now you have to consider the position of the 3-dimensional space. You have gene 1 and gene 2 and the gene 3. And 3 variables to define each individual cells. So you have this cell somewhere 15, 11. So I'm not going to plot them all exactly position, but just give some ideas. So they will be distributed among this in this 3-dimensional space defined by 3 genes. Okay, so once you have that, you also can do a clustering analysis for in the space of 3-dimensional. So that's the idea. And however, if you think you want to take all the information from all expressed gene into account to define the position, to describe the profile of each individual cells, then you have to consider higher dimension, higher than 3. If you have 4 gene, you want to consider 4-dimensional. If you have all up to 11 or up to hundreds of thousands of dimension, you need a way to define the position of these cells in this high-dimensional space. If you go to a dimension such high, and you will end up with a unreasonably defined space which you cannot even think about, and visualize them will be very difficult. So we are going to use the dimension reduction technology to reduce this individual genes description of the cell, but combine some information. For example, those information will be combined into a variable 1, and other information will be combined into another variables V2, and maybe the rest of them combined into V3. So the idea is we will try to reduce, in this case, 11 dimension into 3 newly created variables, and then become a 3D dimensional data, and you can visualize this on computer and paper. So that's the idea of dimension reduction. So this is not a simple collection of representative columns, but we need an algorithm to compute the main components out of this variable space to find the most representative axis, to use them as the variable to describe the diversity and the variation among these data points. Okay, so that's our goal, our analysis purpose of this. So I have slides to indicate, to illustrate again the dimensionality and high dimensional data. How are you going to reduce the dimension? You have a really high dimensional data cloud. You only be able to easily visualize it. If you apply dimension reduction algorithm, you can reduce the dimension reasonably and to a level that you can easily summarize in the clustering and doing this subsequent analysis after you reduce the dimension. Okay, so now I'm going to explain one of the ways that you can use to reduce the dimension. It's pretty easy, and you can consider to construct a distance matrix from your original data matrix. So this is original, that's raw data, original data. And then you want to transform it into a distance matrix. So that's a distance matrix. How can you do it? It's pretty easy. So you consider cell. In this case, if you have n cells and the p genes, so p could be a really big number. So n in single cell RNA-seq data, you have a 2,000, 3,000, up to 10,000. And after doing this distance matrix computation, you will end up with distance matrix with a dimension n by n. Okay, so you have n cells against n cells. You don't care how many genes have been used to describe this n by n, the profile of the cells. So this is a pairwise comparison for each individual cells. Now I'm going to compute the distance between cell 1 against the 2. And then the value be this. And the distance has no direction, then cell 2 to cell 1 will be the same. So this matrix is symmetric. And in the diagonal position of this matrix, it's a distance about the cell compared to itself. So there's no distance, no difference between them. So the distance will be always 0. So, okay, in this way, we have already reduced the dimension, we changed the dimension from original matrix, which is n by p, into an n by n matrix. So let's see how can we reduce the dimension here. So now you see some of the cells, between cells, the distance is large. So cell 1 to cell 5, cell 1, cell 5 have a very different expression profile. At the end, the distance between them is large. And the cell 2 and the cell 6 have a very similar profile, and then the distance between them is small. Again, the matrix is symmetric. So once we have this n by n distance matrix, we are going to use a technique called multi-dimensional scaling analysis to reduce the dimension and plot the relative position of those cells into two-dimensional space. So I will give you an example, see how this multi-dimensional scaling algorithm will work. For example, I gave you a distance matrix between 3 major cities in Taiwan. So we know from Taipei to Taichung, it's 164 kilometers. So in this case, we probably can easily from the distance between plot the distance between these two cities. And then we also know from Tainan to Taichung is 154. So this is 164. And then from here to Tainan. Okay. It's 154. And we also know from Taipei to Tainan is 318. So this is almost linear. So we just reconstruct a map and put the 3 major cities in their relative position on the map from this between city distance matrix. So this procedure is called embedding, and the algorithm you can use is multi-dimensional scaling embedding algorithm. Then you can reconstruct this map. Of course, if you have no idea whether Taipei, Tainan is in the south or the north, you may have a different layout, but this layout will at least satisfy the distance matrix relation. Okay, for more complicated multi-scaling questions, I will use 4 major cities in Australia to illustrate that. We have Sydney, Adelaide, Brisbane, and that's the distance between them, and against Perth. And if you, so this is a little bit more complicated than Taipei, Tainan, Taichung example. But still, if you apply this algorithm, you will be able to put the position of those major cities in the correct place and satisfy those constraints between their distance between cities. You see here the representation of the locations not as the same as their geometric position in real world, but mathematically these two this layout will reflect the pairwise matrix or the distance matrix. This is a more complex problem with more cities. And then again, this just tells you that you can use multi-dimensional scaling to achieve this analysis. So go back to our single-cell RNA-seq data. Imagine you have this big n by n matrix, distance matrix between individual cells. Then you can use MDS to project each individual cells into this two-dimensional space and look at their relative position, you can then cluster cells into groups. Okay, you may ask, how can we compute the distance for given cells and their gene expression profiles? Turns out that that is a complicated problem than you think of. When you compute the distance, we can define the similarity between objects. Then you want to use 1 minus the similarity to get distance. But it's still the same problem. How are you going to define the similarity? In what term you will do that? So this problem is more complicated than you thought. So we can see, okay, the distance computation algorithm is like a black box. You put 2 objects in there, you get a value of the distance between them. Put other object, you get different measurement of distance. So this lecture, I'm not going to go into details of distance computation, and after we define the basic linear algebra key terms, we may come back to see how can you define the distance between cells by their high dimensional gene expression profile data. So we just leave this for now and just think we can easily to get the distance matrix, and you can use multi-scaling dimension to do this embedding. We can also, okay, next I'm going to introduce you a couple of the commonly used dimension reduction algorithm used in the single-cell RNA-seq data analysis. As you have already learned, multi-scaling, multi-dimensional scaling, MDS, can be used to reduce dimension and visualize your cells. So that method is closely related to PCA. It's very, very close to each other mathematically. MDS scaling. And I will also mention tSNE. tSNE stands for T-distributed stochastic neighborhood embedding. This is a very commonly used dimension reduction algorithm. And most recently, UMAP become a little bit popular, but I'm not going to talk too much about this. Instead, I will bring your attention to a new algorithm called the potential of heat diffusion for affinity-based trajectory embedding. So this is a very new, published 2019 paper, I believe. So the aims, the single aim of this reduction is to capture the structure of high-dimensional expression matrix data in as few dimensions as possible to make 3-dimensional or 3-dimensional, 2 or 3-dimensional scatter plots for this big data sets. Okay, so it's pretty much like you have a high-dimensional object. In this case, it's Coke cans, and you want to reduce the dimension. So in this case, it will be 3-dimensional to 2-dimensional. And you can do this in a different way. And for the same object, you will get different presentation in the low dimension space. Again, so they are the same object, but it depends on how are you going to step on it, how are you going to reduce the dimension. Different algorithms may give you different results as embedding of the low dimensional. So don't be surprised if you see different outcomes applied when different algorithms apply to the same set of data. So we are going to show you how I will do the visualization of individual cells. So this is an example from a tSNE plot. Each point here is a cell you generated from the single-cell RNA-seq data. And you're embedding and project the cells on this two-dimensional space. So that's our purpose. So the first algorithm, I'm not going to directly go into tSNE, but I will mention PCA. So PCA is based on the linear algebra, and this is a very mature algorithm which has been here around many years. Try to use PCA to see the hidden structure that's otherwise cannot be seen. It's very intuitively geometrically. So I will give you this example. For example, we are going to sell a teapot like this, and on the website, the online shop. However, the online shop have a very strict restricted rule, and you can only post one picture of your teapot. Then in order to use this offer, you have to carefully select the angle you are going to take that picture to show people, the potential buyers, what the teapots look like. So for most of the people, I think you will select the angle from this side of the teapot instead of from the top or the other angles. So this is by default, you probably will show the potential buyers. The reason you want to choose this angle to take that picture is you want to show the most structure within this data points which create this teapot shape, the representation. So how can you display the most variability among those data points? What you have to do is to rotate your object to lower your dimension to the two-dimensional space, as the picture is two-dimensional. You try to present as much data point as possible that translate to the language of statistics. You try to show the maximized variability among data points. So you create this trajectory, a new trajectory, using a new coordinate system to display the variability among your data points. So you have one data point here, and you have another. So this is almost the longest distance you can find from the data and among the data. So there are endless possibility you can show people using different angles, but this will present the largest variability among data points. So that's the idea of PCA. PCA is analysis, try to learn from your data point and found of this axis. So this is a PCA examples to learn from the data point of three-dimensional and try to find out this new coordinates to show data points using two-dimensional. The option, the selection of this new coordinate system, which is defined by PC1 to give you the layout of your data points and to show the largest variability among them. Okay, so I'm going to use PCA to really create data points with the form a teapot to illustrate the point. Okay, so that's the source code I'm going to use to in the MATLAB environment. So this will read a set of data points, and those data points can be plotted in the three-dimensional space using a scatterplot3 function. Okay, so you have a coordinate number 1, 2, 3. And then after that, we are going to use PCA to learn from these data points and create a new coordinate system. So the information is embedded in the variable C. And then after you project the data points using these new systems, you get those new positions of the picture of your data points of the picture of your teapot. And then you scatterplot two-dimensional, you will found, okay, this PCA automatically found the longest axis and to project your three-dimensional data points into two-dimensional and to show this angle of selection. So this is not rotated by myself, it's automatically detected by PCA algorithm. So I'm going to switch to MATLAB and see how this can be run on there. Okay, so what you can see is my MATLAB environment, and I'm going to read that data points in. So let me clean my workspace. So you see there's nothing in the workspace now. But once I load this teapot information, all the data points are stored in this variable called vertices. And there are 4,608 data points, and each data point is defined by 3 values, their dimension. So that's what it is. Okay, so this is a three-dimensional object. So we can use scatterplot to visualize the position defined in this variable. In the this is carefully designed, so this is a teapot. Okay, the teapot. Now our project is to visualize using one picture, two-dimensional, either this way or this way. This way, there are endless possibility to show this in a two-dimensional. Okay, so I will let PCA to choose. So this is a PCA function, built-in function in the, it's a function in the statistic toolbox of the MATLAB. So this can give you the coordinates of the new system, because we are going to view the teapot defined by this coordinate system. So in this X, Y, Z, basically, the direction. And S, you see S is the same dimension, that is after rotation, the new position of the data points. So I'm going to show only the first two dimensions of S. Because with lower dimension, from 3 to 2, and then the first 2 that major PCAs. And that's the outcome of this PCA analysis. You see, the teapot have been carefully rotated and projected, and the first 2 PCs will display the most variability among those data points. Okay, I'm going to switch back to our teapot presentation. I'm not going to go into the details of mathematics and linear algebra about the PCA. Next lecture, I will start from the matrix multiplication and maybe eigenvalue definitions, and then you will have a better understanding of PCA. It's such an important algorithm. But for now, I'm going to introduce you different dimension reduction algorithm, only ideas. So for the same set of data, and if you use the PCA, you may generate an outcome like this. Okay, so each data point maybe is a cell, and however, if you use tSNE for the same set of the data, exactly the same input matrix, you will get different layout of cells. Why people using this more compared to PCA? One of the reasons is you get better resolution of the clouded data points. See here, there are a lot of cells, data points being put onto each other, even in a very small area of space. It's very crowded. However, tSNE will give you a representation which is much more spread out. And the reason for this is I will illustrate using this London underground map. So this is a map you will see in the underground station, but now at this moment it's a real distance between each different stations. People trying to do this like to do this transformation in order to make those crowded data points more spread out, so you can easily to see the information, the relationship between stations. So same idea applied to single cell analysis. In the use of PCA, you will get very crowded layout of your cells. However, you want to do the transformation to spread them out. The same idea as this underground map. So the transformation is done by the tSNE. Okay. So again, to show you the difference, you have a very crowded, super crowded cloud of data points, cells that cannot be separated by PCA, but they are much better embedded independently and more spread out by tSNE. So tSNE is one of the most commonly used approach to visualize your single-cell RNA-seq data. As I said, most recently, we have a new algorithm called PHATE. And for the same set of, exactly the same set of data, PHATE creates a structure, two-dimensional representation of your cell like this. There are some advantage of this. It tells you the elongated structure among the cells. So some of the cells have very special profile compared to majority of them. the other cell types, and it creates this one arm, which you cannot see those subset of cells in tSNE or PCA. They're very useful because later we will mention about the pseudotime trajectory analysis. So you can build the trajectory among your cell to illustrate the cell which had different development stage, for example, but you just cannot create those trajectory among cells which represented using using the tSNE coordinates. So there are some very well suited field for PHATE to be applied. Again, you see you can use a different visualization algorithm and get different results, even though your input is the same. PHATE can also do 3-dimensional. So I'm going to mention about the PHATE in a little bit details, and the whole idea is based on the machine manifold learning in idea. So the manifold is a mathematical structure, and you can imagine that a structure can be used to describe very complex relationship among data points. In this case, you see if you have a global view of where the data points are distributed, you will have this structure here. However, if you compare, say, 2 data points, one is from here and another here, and if you don't have this manifold structure on your mind with a global distribution of your data point, you only look at the difference, the distance between the 2 data points. They look very close to each other. So this is a Euclidean distance defined by between these 2 data points. Let's see, A and B. However, if you treat the whole distribution of data points as a manifold structure, then from A to B, you have to go through the manifold structure defined paths. So you have to go, say, from here. Let me use different color. You have to go from this position along the trajectory to be in this position. So it's much longer distance compared to Euclidean distance you naively defined based on their original position without looking at the global distribution of the data. So how can manifold learning process to learn this structure? So idea is you are going to build so-called KNN, K nearest neighbor network. So you compute KNN structure, and you then only link them if they're close to each other. You define a number like a K. If you define K with like 5, then you only found the 5 nearest neighbors of them and connect to that. So in that case, you create KNN, the nearest neighbors, and you learn from the network about the structure. We will have time to go into detail of the KNN in some lectures later. And then for this PHATE algorithm, exactly doing this, and then use a kernel approach to transform the length, the distance between cells. So here you learn from the KNN, and then you learn this structure. So this display can help you a lot of downstream analysis, such as the pseudo-time analysis, clustering, and also applying the noise and the visualization as well. So I will go into some details of this algorithm. This published 2019 in Nature Biotechnology, and by a group from Yale, SNE group. So first, they construct artificial data points, and the structure among data points is a tree-like. And then use PCA, you just cannot recover the structure of the original. tSNE play a slightly better outcomes and the PHATE will reconstruct the whole original structure of the data. So this example just justified to use PHATE to understand the structure of the data. Then they apply to the real single-cell RNA-seq data, and then that's a PCA result, a tSNE result, and then they demonstrate that PHATE will capture the development stage and the transition of the cells from one stage to another much better than other visualization algorithms. So that's the main steps in PHATE algorithm. We start from the matrix of the gene expression level of single cells. So that's exactly the single cell RNA-seq data UMI count. And then first compute the pairwise distance, and then transform the distance using the kernel method. And then kernel method will define the affinity, and learn from the global relationship, use the diffusion process, so the KN is involved here, and then from this relationship, and then transform the distance again. At the last step, you will have an embedding algorithm, which is MDS. Remember, MDS is multidimensional scaling. Start from a distance matrix. So if you jump from a distance matrix, step 1 directly to step 5, that's standard MDS. But in the middle, those steps are learning the structure of data and transform the distance, changes value basically, and then give you the results. But still, at the end, it's MDS itself. So this illustrates the main steps. So you have your input single-cell RNA-seq data, and they assume they are in a very complex relationship among those data points. So this is the structure of the manifold. Then you start to compute pairwise distance between all the cells. So cell I to J, compute every pairs. And then you use this kernel function to change your distance to affinity. Then there's a stochastic process simulating the diffusion, and then further transform your affinity into another distance. So that's the distance. After this, you have a remember, you have, at the very beginning, you have a distance, but after this transformation, still you have a distance between them, okay, after this treatment. Then the last step is to use MDS to present your data points in 3-dimensional or 2-dimensional space. So that's the information, the explanation of each steps from the original paper. All right, that's all for today. And next lecture, we are going to move into clustering analysis. And before that, I will introduce some basic concept in the lineage algebra, so you will have a much better understanding of the PCA and also the rotate of the coordinate system, and also projection of data points from one coordinate system to another. All right, thank you for your attention, and that concludes our